Good morning, everyone. It's my great pleasure to welcome Professor Siwei Wang from Stony Brook University, New York. And Siwei is also a affiliate member of NITMB, and she's actually a Chicago alumni. She graduated with a PhD in computer science, and after that, she switched her interest to neuroscience, like computational neuroscience and theoretical neuroscience. And she did a postdoc fellow at Max Center for Sensory to Action. And then she moved to Chicago again in the Palmer's lab. After that, she got like a professor job at Stony Brook University. And her research interest is in information theory and using information theory and machine learning techniques and combine that with neuroscience. And with that, I like to welcome Sue. Let's give her like a round of applause. Oh, wait.
Thank you. So I moved to this professor job about a year and a half ago, and you will see three parts of the talk. The first part for the perma lab, maybe you guys still get some of the familiarity. So that come from my postdoc work. And the second part, I would introduce some of the ongoing new collaboration with Greg Schwartz from the Western University. And third, I would introduce some kind of a new mathematical framework we are just ironing out to investigate natural behavior, and that's based on cross-species comparison of monkey animals. And, before I introduce all the math, I want to give you a little bit of experience why we think evolution occur in the natural environment and considering natural environment itself is important. And this is one of my favorite incentivize for exciting audiences when I'm giving a talk, especially I don't know the background yet. So it is a coral reef, and showing the cephalopod here is an example cuttlefish trying to blend into the external environment. As we go through this video, you will notice that the cuttlefish is trying to change the shape, color, and texture of its skin or exterior appearance to blend in. And what's available in this external environment supports the camouflage capability and sculpted what they can be doing throughout the time scale. And so far, this video is showing you a correlation. That is, when we see this complexity available in the external environment, we are seeing all kinds of different changes. But what is this the actual cause, or is this the fundamental equipped computation in the brain? Can we have unlimited range? So the second part is we're going to do something that probably a little bit unpleasant for this cuttlefish herself here. We drop them into an artificial environment, and then we ask them to camouflage. And in this environment, immediately you want to observe that there's a checkerboard that's artificial, that's not real, and then the cuttlefish naively initially trying to blend in. And this is actually a BBC documentary made to show testing the capability of the animal. So the anchor of that is the Dr. Roger Hannon, he has been working on this camouflage phenomenon for 30 years or so. Although we would call this an amazing cuttlefish camouflage, all of you are going to agree that animal going to die if this is the environment and living this way. And if this happens, we won't see camouflage or we won't see cuttlefish today. They stop contributing to the evolution diversity. So for that very reason, they would move to the lounge, which has the visual feature that fits or that has some kind of naturalistic feature characteristic. And you can also observe the difference of animal maneuver between the checkerboard and this granular lounge feature. Within this blend-in, while it's more similar, the animal also does not jitter. It's resting into this environment. So that actually tells you the animal sensing its blending is good enough. So this is just a very quick experience to tell you understanding the real complexity of the natural behavior is important for us to gauge how the evolution sculpted the neural computation. And that's we don't not only observe that in the biological or in the natural world, in human society, this also applies. So one famous example is the education opportunity equality events. When the first analysis of this equality only used US data, remember United States for so many years up to today is still the richest country of the world. The finding is that student achievement is not correlated with the school quality. But 16 or 14 years later, if we include all of the countries around the world, we completely turn the conclusion upside down. That given all of the diversity of economic status, it is important for us to consider the school quality and the economic status. So the real complexity throughout all of the scale is necessary for us to find the correct conclusion. And now I'm going to give you a little bit of an experience why actually working on real complexity is challenging. So before I show you next slides, I would invite all of you from where you are, please take a look at the center of the screen. And other than Jared, all of you probably didn't see this picture before. So there is a blending hidden animal there. And given all of given the data we analyzed, we know if you're an expert in spotting the camouflage, you will be able to spot it in 3 seconds. So I'll count 3. And if you find it, raise your hand. Okay. Who found the animal? So there's no wrong answer. And if somebody has visited Okinawa, I could say you can find it in one gaze. Because this is taken by the seashore of Okinawa. So I completely fail upside down when I give this talk to Japanese cohort. but none of you have been scuba diving along the shore. It can be difficult. So now, anybody found the cuttlefish? May I ask one of you? It's okay if you guess wrong. May I ask one of you to try to spot or guess where it could be? Would you mind pointing it?
At the back over
And the back means where?
at the if you keep going,
Yeah.
the top there.
Yes, you're right. That's the cuttlefish. Okay. So for this people sitting here, this is where the animal hidden. So as I pointed out, you cannot unseen it. Okay, so if we have the simplest possible model to model your decision process of this perceptual decision making, and one of the classic is this guy, the diffusion decision model. So our success, the expert, you start to gain experience, is accumulating evidence all the way, although there are time of hesitation, it still reached the threshold of finding the cuttlefish. And a lot of you who didn't find it follows the green curve. You might see something similar, but by the end you said, okay, I cannot find it. And I intentionally put the red curve here because this picture was taken by Dr. Hannon himself. And he can spot cuttlefish, the thousands of cuttlefish we tested with one gaze. So we have to remove his data and everybody from his team from the distribution because they skew everything so badly, given the slope. But even if you're able to find the cuttlefish, if I ask you the question, what is the feature or what are the sequence of characteristics that promotes you to find the cuttlefish? It's probably still difficult for you to describe it. And that promoting of the description is one layer of difficulty. That is, we have richness of all the repertoire of possible natural image. And the second thing is the decision making when we are looking for things, it depends on how long we make the right decision. Or said another way, a good decision means we need to achieve the success within relevant short amount of time. And in mission critical context, that is a good decision means survival or death. And then such a decision-making sculpts neural circuits alongside the entire sensory to behavior arc. And so in the first part of the talk, we'll go through how we model neural computation within the two types of survival critical behavior. One is the salamander catching the prey, and the other is the fly escape from the threat. And the common computation we propose here is because of motto, brain works on single trial. And what I meant by that is when we are processing information, as we wander the world, we are always sitting in a continuum stream of information and trying to make sense from our past experience and decide what component from our past experience is important to guide our future action. And it's in these kind of simple animal model, when we are able to coin down the temporal constraint within this kind of predictive environment, we're able to put into a mathematical framework to make prediction. So I guess, so all of you, let me test it. All of you have done or understand what mutual information is? Raise your hand or no? Okay, all of you. That's good. And so for us to do this, the model-free framework we propose, or we try to use as an example here, is the so-called predictive information bottleneck. And that's very simple. It is just describing what I said to put them into a constraint optimization objective. How much I know, or how much I encode from the past, that's the I past from the stimulus, and how much of that I can turn them to be utility, to be useful about my future. And you will see in these two examples, because of the stringent time constraint we put in this animal and for their success of their survival relevant behavior, we are able to point even at the early sensory layer, this type of survival constraint is sculpting how much they encode from their previous external environment, how much of that is predicting the future state of their environment. And this constraint optimization actually sprouts from the very well-known main school hypothesis of coding for neural system, that's efficient coding. It started by the Shannon's information theory back to the late 40s. And if we turn that information theory essentially is saying, and given we know we are building an encoding channel with enough source coding capability, that's the mutual information, big string X and Y, we would like to minimize the distortion. And the dual problem actually becomes, we want to maximize the encoding of the source by keeping the divergence within a certain level. And this particular hypothesis, first populated by Atif and Barlow in 1961, only 20 years later, we found actual evidence in the first layer of fly motor neuron. So in the one of the fly neurons sensing the contrast, that's the dark and light of the external environment, we notice that that particular neuron assign its response range according to the distribution of contrast condition of the natural environment. So this is maximizing how much that neuron can encode this contrast information. However, if we start to reapply the same principle going into deeper system or going into guiding behavior, we know there is a bandwidth shift difference. So for this particular neuron, they are following up work from the same group saying that they are encoding the bit rate of this neuron is about 100,000 bits per second. But when you're doing eliciting behavior for mouse, the scale of behavior relevant information goes down to tens of bits. So why is this bottleneck? We propose this bottleneck generally coming from metabolic cost. Spiking is a very expensive activity. From the same author, Simon Laughlin, in 1986, it says in fly visual system, one spike would cost about tens of thousands of ATP units. And in the extremely nerdy case, we can calculate if they're sending out one of the stimuli, how many bottles of Gatorade we need to feed fly so they can spike enough to send the information. And for that very reason, we want to put that not only in we want to put that into a constrained scenario, say metabolic cost is the bottleneck, how much information can be used, and then we're gonna make the best use of it. An information bottleneck method is simply a mathematical formalism that unifies the notion of the cost and the benefit both into the mutual information theory term. So we are essentially doing a cost-benefit trade-off analysis, and we formulate both the trade-off cost and benefit into mutual information. And from the mathematics sense, we are using from mathematics sense, this gives us the flexibility to not make assumption of what are the possible mechanisms, so we can locate what's the optimal mechanism, as you can see later example. And from the operating perspective, this is one of the general way. If we do not exactly know what decoding function is, this can give you a good estimate of what are possible. So we can we'll explain its connection to Bayesian optimality. So we can see that when we are maximizing the channel that's maximizing the mutual information, we are also optimizing when we're decoding the Bayesian, the decoder should approximate to the near optimal Bayesian decoder. And information bottleneck, although it was first proposed by the early 2000s, the first actual evidence in neuroscience of it actually can be used to explain neural coding was by Stephanie Palmer in PNAS in 2015. And there I'm showing you this bottleneck of salamander retina. That is saying that the spiking activity of the salamander retina from the past state is predictive enough to the future state when they're viewing in the external stimuli. At the same time, Tali Tishbit's group from the Hebrew University of Jerusalem did a very similar predictive experiment in the audible paradigm. That is, they're playing a sequence of stimuli with two different varieties. And one of them has a much higher occurrence frequency, and the other is the audible, that is the surprise stimuli with a very low frequency. And as they're playing the stimuli, what they notice that in mouse auditory cortex, given there is a less frequent and more frequent frequency imbalance, the auditory cortex neural activity also start to remember the past sequence and making inference to guess what's the future identity of the stimuli, especially if we're holding off this audible sequence before the animal figure out, okay, these two stimuli are actually independent. So now this comes to how we make how we discover principle into neural system. What we are looking for, or what computational neuroscientists like to see, is one principle gets implemented repetitively by two systems going through independent evolution. And that means it's possible this is a common computation mechanism that transcends species. And not surprisingly, now if you look for the open the wiki page of audible paradigm and prediction, you will see that this work not only found in the auditory cortex of mouse, it's also found in human P300 EEG data. So our auditory cortex also does similar predictive computation. And now we have pointed down that prediction is important for sensory processing. I'm going to show you how we use that to discover, to have a more deeper mechanistic understanding of neural coding. So the particular format of the first example I'm showing you is in this format of we are maximizing what information we encode about the future state. And when we are doing this, we essentially hold the past constant. The encoding from the past constant is an adjustment. We're taking the neural population we recorded from as is, we don't change them. And the optimality here, I need to has several constraints or several notions we need to bring up before we introduce the experiment. First, how much information available for prediction is determined by the problem structure. That is it's independent from the processing itself. It's just the general correlation between past stimulus and the future stimulus. And second, the optimum here in the phase plane, this curve I'm showing you, as long as we fixed x-axis, as we trace the intercept of the future, this predictive information is the maximum or is the optimal amount of predictive information available for this given cost of information we encoded from the past. What does that mean? Two things. First, every point of this curve is the optimal solution, given the exact past information we encoded. And second, every point at the curve is a unique solution. If we find a neural system operating close to that bound, there's no other way they can encode this information. So that makes us that gives us an anchor to perturb the neural circuit or perturb the stimuli to see why if we observe some near optimal mechanism, why they're happening. So we're gonna use this particular way of investigating near optimality of neural coding to look at the rapid behavior of a fly. And why is that interesting? Fly can escape a visually guided threat within 40 milliseconds. And for humans, we blink our eyes. One eye blink is about 100 milliseconds. And that's why there's no YouTube video you can actually see you swat a free flying fly unless you control them. So the fly actually turn away their direction half the time we even blink us. And another reason fly is fascinating for mathematics is they have this type of clean compression. We know fly has compound eyes, and there are hundreds to thousands of compound local motion detector determined by the body size of the fly. So, and all of them all compressed into 6 to 10 global motion sensing neuron, again, depending on the body size of the fly. And the second, fly follow very striking scaling law. So for all of the global motion neuron, as you see, I plotted the dendritic arbor here. The arbor size and span also depend on the body size, but their shape and their distribution follow analogously according to small and big eyes.
If I blind the fly and then I try to slide, will it still escape? Right? Does it just use the visual system? It seems like it might sense pressure or changes in the
airflow. Yeah, you made a good point. Hot air and airflow gyroscopy will still sense that. And airflow is slower than visual for this particular processing. Yeah. And the reason we are able to make a mechanistic prediction is not only because of the information theory I introduced, it is also because of this experiment technique that's able to perturb or that's able to investigate the synaptic connectivity. So on the left-hand side of the slide, I'm showing you a dual recording of intracellular recording. That is actually, we know there is a synaptic connection between these two neurons. And then we're injecting current from one neuron and observing the activity change in a different neuron. And here we are evaluating how strong it is of the synaptic connectivity. And this actually allows us for this type of small circuit to build a compartmental biophysical realistic circuit that's showing you from the right. And in this circuit, we not only know all of the single neuron electrophysiology property, we also know that the connectivity among the neuron occur at their axon. That's the compartment sending out the output. And also they are connected through a subpopulation readout. So the green spot telling you, although there are 20 neurons and 10 neurons for each side and 20 neurons both sides, they're only read out from the center 3. So there is a redundant coding there we would like to discuss. We would like to find out why or what the benefit of this. And the key component for the wiring, now we see that it's just hinged on this gap junction. And that's what's interesting. Gap junction is different from the general biology class of chemical synapses. It doesn't make neurotransmitters. And it's also not one direction. Gap junction is saying your two neurons need to be very close to each other. And by very closing, they're making the bidirectional channel to allow the current to flow through. So the benefit of the gap junction or electrical synapses is is that right? 100 times faster? Okay, thanks. It's much faster than the chemical synapse. And the downside of that is it's really hard to see in electron microscopy. So it's only, and although there are 5% of synapses in your human brain as gap junction, we're still in a slow process of discovering now. And now we have a behavior of escape. We have a circuit we know responsible to processing the behavior. And why do we think gap junction is the reason that's controlling the successful failure of that? This is because, again, of the time scale. So this is a video of the flight turning away from a threat. And the video's original frame rate is 7,500 frames per second. I slowed it down 500 times so you can see them. They can turn their wing beat about 3 wind bit to change the direction. And that's 40 milliseconds. And within this entire time course of the flight sensing the escape, I would like to point out that, let me go back. I would point out that this flight is escaping its threat by executing a rotation or a turn. We call that bank turn. So that's in your water and air show. If you see two airplane do dogfight, that's how they actually encounter themselves and split away. They need to execute all of the rotation with all of the possible 3 body axes. By that rotation, its rotation speed can be up to 6,000 degrees per second. But the trade-off it pays is it's highly unstable. So the eco motion must be controlled at real time. However, the visual stimuli happens way before we are able to have any feedback that's showing in the green spot. So hot air control, the gyroscopic control wasn't available in half of the course of this particular flight maneuver. So the visual prediction has to come into play. And this is the whole setup. We put together the recording or the experiment availability of a biorealistic circuit, identify the exact mechanism we're studying, and then framing the problem given the behavioral constraints. And now we're going back to the information bottleneck. We will use this framework to ask, if we have the system, we are able to perturb the wiring diagram. How much does the wiring diagram change the encoding of the behavior relevant information? And how much does the readout mechanism change or help the readout of this behavior relevant information? So as I showed earlier, now we want to see the neural system encode close to the bound. And we're exactly seeing this as both the subpopulation and the entire circuit is existing close to the bound. So they are near optimal. And not only they are near optimal because of how we are solving, like we can do how we are doing the calculus of variation given the information bottleneck methods. If we rewrite this both of the mutual information term into their conditional probability term, we can actually do the calculus variation over the PZX. And here we can discover what features are available under the hood of this near optimal solution. So that means we have different features of the information bottleneck, and different feature aggregately adds up to contribute to the predictive information. And feature with more information will be coded earlier than a feature with less information that corresponding to different critical points in this information curve. And this is exactly what we approximated with the variational inference of lower bound. And so in this plot, I'm showing you 4 different steering directions the flight is trying to differentiate where they're going. And these 4 are only different about 10 degrees each. And the scatter plot on the left versus the right is just whether we're using the entire population or using the subpopulation. And we're seeing the same discriminability, just the reformatting of the manifold while maintaining the discriminability. So it's a compressed representation, keeping all of the information behaviorally relevant and still simplify it. And this is the one angle of information. When we are maximizing predictive info, we are able to explain when the system is optimal, what does different elements of the neural coding, the population pattern, the wiring mechanism contribute to this particular predictive coding regime? And if we turn the table around and just say, we know what happens, and now we're just trying to simplify what's possible to encoding. This is the ongoing collaboration with all of your favorite, James Fitzgerald, a couple of years ago. And we are now saying, we know the flight can do direction selectivity. And what's the simplest possible encoding channel? So we turn the channel coding scenario to be able to have that capacity for direction selectivity. And here I'm showing you, turn the table around using a dual formal information bottleneck. We are able to predict how many direction selective cell types are there in the early flight visual system. And this is this is one level of investigation when our experiment provide us very intimate understanding of the one-year diagram and the cell types. If we move on to a more complex system, a lot of times we don't have such a detailed knowledge. Like one example is when we use UTAR array and only patch into the intersystem on the dish, and this UTAR array can record a ton of neurons, and we can only get their spiking activity, but we lose the connectivity between them because different electrodes are far away between each other. And also we can only determine their activity once they have spikes. So we don't get those greater signal connected by the conductors. But we gain by being able to see neural population activity at a larger scale. So this is the next one I'm going to show you. That's population prediction in the salamander retina. And in the first work of showing prediction at works at salamander retina, there was a demonstration of a small group of retina ganglion cell encodes efficient near optimal prediction to bar stimuli. And the bar stimuli means we are able to control the dynamic as an over-damping oscillator. So we actually can write down in closed form what kind of predictive components are. But what happens in natural stimuli? A natural movie is, and what happened in the large scale? So there are different complexity for natural stimuli. One is the encoding, the other is the stimuli itself. And because of this, when we are solving the problem of investigating population code of the natural stimuli, we're not going to try to unpack as we did before. Instead, we will parameterize the encoding process as a deeper neural network. And that brings us to another mathematical property of this information bottleneck solution. So every point of this black solid curve, while they serve as a unique solution, together they are the convex hull of the entire solution space. So once we are able to approximate the predictive information, that's the dashed line hits the end, we're able to approximate available predictive information. All of the curve it traces any of our encoder, how it traces or how the dynamic emerges to encode the predictive information must be sitting below of this or within this convex hull, and that's a near optimal curve. And with this particular way of looking at the encoding, we're going to investigate how salamander retina encodes 3 different natural movies. And we chose this with ecological value. That's because salamander is a very old animal in terms of its genomic, and it has a very interesting life stage shifts. We're showing 3 movies. One is underwater, the other is in the moving leaves, and the second and third thing is the waves. The reason is these corresponding to the life change, corresponding to different life changes salamander. When salamander, when salamander was a teenager, they would live underwater. And when they become adult, they live at the land. So within the salamander's lifespan, there is a transfer learning problem. They need to find food after they move to land. Based on the experience, they learn how to find food underwater. And they're not going to go back to live on the land when they hit puberty. So how did they do that? Well, how did they do that means how does the retina does that for in this particular setting? And now we goes to why we use deep neural network to approximate the encoding instead of enumerating or doing the closed form investigation of those. So when I'm teaching intro to neural computation, we always start with showing the different orientation selectivity or different activation by the grading stimuli. And the reason is we are able to enumerate all the possible discrete states of the stimuli. What's the variability there is fully understood. That's not the case if you have a natural stimuli. Likely for different states of a natural stimuli, a naive way is we need to enumerate all of the sequences. And that very quickly give you combinatorial explosion. We cannot use that closed form investigation. And second is not only the stimuli itself is complex, the response is also complex. And this is an experiment of a single neuron, comparing the single neuron response from the primary visual cortex to bar stimuli and natural stimuli across different trials. With the bar stimuli, as you could see, that neuron fire consistently over trial, and with natural stimuli, that response fluctuates. So in neuroscience, this is a very well-known effect, not only in primary visual cortex, it's everywhere, called representational drift. But this one actually tells you, even the brain, when they're encountering a natural condition, they behave differently compared to the lab condition. And we would like to deal with the real complexity. So how do we set it up? We have 93 cells are the ganglion cells, and luckily 82 of them are the same cell type. You'll see why cell type matter in the third part of the talk. And second, we have a 20-second long presentation of the natural movie. And here is the aggregated receptive field. So we know these retina are going to be responsive to all of the heat map conjecture. And we're building the encoder of saying how much does these retina spikes encode the natural stimuli, encode this natural movie. And each movie, of course, we build one decoder. And the decoding problem here is, can we reconstruct movie frame from the mean firing rate of the large retina population? And we use mean firing rate here exactly also because it's a sensory experiment. Every external variable of every single trial is hold consistent. So the variability across trial coming from the neural population itself. And in the behavior paradigm, if you do single trials throwing a basketball as such, the behavior variability would go into the external variable, and you will need to change the current paradigm. And the architecture we use is this particular encoder decoder. And there are two things I would point out here. First, the encoder is only taking the input as a neural spike train. And the movie frame only participate the decoder parts through the loss function of what we predict compared to what's the true frame. There's no movie frame in the stream of the input. So this is actually reconstructing what the retina know about the natural scene. And second, we are impinging latent representation across different layers of this encoder as we go through the spatial temporal compression and trying to extract a hierarchical representation of the natural movie. So third is we are by training this encoder decoder to a high level and matching of the ground truth. Now we can open this trained model, look at the latent representation to understand if we at the same time when we are replicating the retina encoding process, do we pick up any feature that's similar or that correlate to known retina computation mechanism? And here we are only making correlation argument just because the same thing, the non-model is not there for us to make a causation argument. So the first evidence is there are something interesting here. So this is the 4 top layer activated feature. And if you pay attention to the lower left feature, you will agree to me that we remove the flickering background. I'll play that again. Sorry. So the top right, the bottom right feature, we remove the background. And that removing the background part actually cascade across all of the layer as we go through different spatial temporal scale. And so in the deeper layer, we're seeing that a lot more clear. That is, we trace all of the movement while we leave the background apart. And that's adhered to what Greg Schwartz's group found a couple years ago about the foreground background separation. And this type of qualitative finding suggests to us that the encoding itself is tracing feature changes across the scene. And can we actually tell how the different feature encoded across different scenes or differentiating different frames? And that is this the second exercise of quantitative encoding. We are trying to decode different frames. We're trying to ask the decoder where it is in the movie as a proxy of what the natural feature. Yeah.
I was clear. So so in this foreground background, they're separated in this latent space. So in the
Say again, sorry.
In this foreground background,
yeah,
they separated in this latent space.
They're separated in the latent space. Yeah. What we had those features there is we essentially just pick out the activation of the latent space projected to the filter and visualize that filter. So that's the separation in the latent feature itself. Okay, so this decoder is playing the exercise of we're choosing how many latent feature we included in the latent decoder and how well they can help us to differentiate different frames or differentiate where we are in the natural movie. And given all the motion content, we know that differentiating single frame of the natural movie is tracing the change of stimulus feature. And we can see that only using 5 features, we are almost able to achieve perfect decoding, while the chance level of this decoding is only 1%. And this is not the most surprising result. The most surprising result is we fixed the decoder that's only trained on the spike train decoding or viewing fish movie. And we use that trained encoder and get the activation from spike train of leaf or water movie and do the same decoding. We can still get nearly perfect decoding performance by adding a couple more latent features. This is saying the retina encoding or this type of decoder is figuring out what is the common low-level features among these natural things and extracting them into the latent space. And this actually tells us that this extracting out allows us to develop a proxy of natural movie features. And later we're using that to understand, interrogate the encoding mechanism, why such a low D is effective. And the reason we're able to do this is coming from this. We can use this very simple example to tell you why. First, this is given the visualization of the latent space. We notice that there are 2 different possibilities that we can tell these 2 frames are different. The first one is a textural difference. The fish is in the different position of the frame. The second one is a motion difference. The fish has the same stripes across the scene at a different time, but they need to sense the motion progression to know the second part is different from the first part. And so that suggests to us at least there are 2 different streams of stimulus feature. One is the texture and the other is the optic flow, or that's the motion. And that's exactly what we noticed. If we cluster our latent space, we actually should start to notice that the latent space positions similar texture feature and similar dynamic motion feature into similar areas. And that allows us to find out how many of the features are emerging, how many discrete states are emerging when this retina is encoding the natural movie. And this allows us to perform the predictive information IB analysis based on this linear optimal solution as I proposed earlier. Once we have near optimal decoding performance, what is the underlying low dimensional representation? And that's the question we answer here. And what we found is, again, interesting and help us to have some new interpretation. We actually know there is a low dimensional hypothesis that neural code has intrinsic dimension. But what's the benefit to have a low dimensional representation? Here we found if we simply add the encoding of this marginal distribution from the static and the dynamic stream together, we are encoding actually less information compared to we collectively encode the joint distribution when the encoding space locate within the low dimensional regime. That is saying if the neural population or population code is keeping a low dimensional, it can choose the synergistic coding regime to acquire more information about the natural scene together versus coding separate information from separate medium. And also, this compression is much lower compared to what we know for the video compression. And all of this compression, given that we are achieving single frame discrimination rate, and it is keeping all the available behavior relevant information in this movie. So now we already found I gave you 2 stories, both of them telling you predictive information is important in biology, or information bottleneck in the predictive context can help us understand neural computation. And if we strip away the biological context, what does that tell us about the data processing? So in general, information bottleneck methods, we essentially is working on an encoding problem that we have a complexity of the channel. That's what we encode from the input, and the relevancy term, that is what we care about the variable. And under the hood of this objective, we're assuming a conditional independence. That is, we hope in the optimal form, our encoding or our compressed representation Z only contain component about Y available in X. This is when we have a This is when we have a causal chain that Z come from X and encoding Y. However, let's take a step back. Let's take a step back and relax that constraint a little bit. So we know that so we can relax the condition to be a symmetrical form and to say X and Z are only related because of Y. This relation makes them bidirectional instead of one direction. And this relationship allows us to write it into different form. So we can say both X and Z are parameterization of Y. And whether they're related or not, depending on whether we use G based on YF or not. And bear with my note. I'm going to call it Z1 and Z2 as a simplification. You'll see why in the next two slides. So now the XZ, the complexity term in the above, if we rewrite the mutual information in this expectation and distribution form, you can see that this is just the expected performance if we're using a Bayesian representation encoder. And here we are showing the sample-based form. That is, the above term is based on we are able to evaluate this equation or this mutual information of the entire distribution, which in realistic scenario is not possible. Generally, we only have access to part of it, or in machine learning term, batch of that. So given the size of the sample, this is sample-based approximation of the mutual information, regulated or constrained from above of how many sample we have access at once. And given that, given the intrinsic connection of when we are able to achieve the mutual information and the Bayesian optimality, this is actually the upper bound of any possible decoder, including sample-based self-supervised learning. So now we build from the mutual information as I reversed the calculation of the self-supervised contrast learning, how they connect to mutual information, and just showing you we can also build a way backward to say mutual information is the upper bound to any decoder, and we can actually use that to explain how self-supervised learning works. And this Z1, Z2 can be in many different scenarios. First, when you're doing deep neural network studies, they can be different stream of augmentation. Second, different architecture. Third, different training paradigm. So how does that help us? In the first example we're going to talk about, it helps us by combining together with another topological effect we know when we are able to train the influency loss or contrastive loss into its near optimality. Optimality does exist, and that was happened. And we know that from David Donahoe's seminal paper in PNAS in 2021. That is saying for classification task, if you train the encoder optimally, that means your training accuracy is 100%, and you stop increasing your test accuracy. We are seeing that geometrically, all of the samples belonging to the same class start to push into a distinct cluster, and the relationship between each of the cluster form an equiangular tight frame. That is, they have maximized distance among others and maximized angle. And what does that actually mean topologically? When everything is maximized and simultaneously satisfied with C class, with multiple classes. Think about, imagine that they have 1,000 class, and we need this kind of lattice occur in that 1,000 class or in the C-1 space. The dimension, effective dimension of packing such an ETF, such a clustering structure is C-1, one of the cluster. And also there's nowhere else one of the cluster can go. You can prove by contradiction that that's the only space we can collectively achieve the equiangular frame. So this type of clustering structure formulate a topological set that the clustering distribution or the near optimum encoder pushes all of the sample to the boundary of a topological set that covers the C-dimensional metric space. And this coverage allows us to use this clustering effect to find the solution for the IB problem and perform striking dimensionality reduction. And given the time sake, I will give you an example. And so the example I'm showing you is based on CIFAR 10, which is 10 different classes of the natural images. And there are two questions here. I need a volunteer to take a guess. The first one is, after you take a look at the 10 different classes, which one you think is the easiest to tell?
Ship?
Oh, you know that, yeah.
No, I was just guessing. I didn't know that.
And which two are the hardest to separate?
Bird.
Bird and what? Two. So you should give me two. Cat? Bird and cat? Any other answers?
I was saying cat and dog.
Cat and dog.
Automobile and truck? Yeah.
Automobile and truck. Okay. So the next. So we are, given the constraint I showed you, we're able to recover 10 different features out of the 2000 dimension space of a pre-trained retina 50. Training to up to near optimal. That means the testing accuracy is 95% for cipher 10 test. That's this SOTA result for this architecture. And now I'm going to show you by adding IB feature according to how much they contribute to classification, the answer of the two questions I told you. So the first one, sorry. The first one, the ship does hit 93% if we use only two features. So Greg is right. And the second is, as we cascade along this hierarchy, the last two features dedicated to classify cat and dog. So Jay is right again. And also the interesting part for this type of information hierarchy is decision-making exists in the subspace. What do I mean by that? When ship achieves 93% there, all of the correctly classified samples would consistently remain correctly classified throughout the feature hierarchy. And then the rest of the decision process is just on those one that was confused in the lower dimensional representation. So now we can actually use this to develop explainable feature representation for data we don't have access to what meaningful features are. And in the rest of the time, I'm going to show you one of the very exciting new work that we actually is working in collaboration with Greg Schwartz on the cell type classification. And this is interesting in data science as well. And also because of long history of we have been using the intracellular recording. So intracellular recording is the fundamental way for us to probe the cellular activity and also cellular diversity. We do that by different steps of current injection. And from the current injection, we see all of the different voltage traces sampled at super high frequency of 20 to 50 kHz. And then because of this type of multiple sample experiment paradigm, the experiment itself cannot be too high throughput. And so even the biggest data set published by Allen Institute contain at most 1900 cells, and that's about a couple of years of their effort. And now, for electrophysiology or for all of our neuroscience textbook, we take the voltage trace, we actually do not look at them. From computer, if I'm just taking the beginning of neurocomputational neuroscience class, I would remember resting potential, sag ratio, and all of these biophysics features. And that's in the data science paradigm that we are doing intentionally dimensionality reduction from this high resolution space to global feature. And the reason is simple. Given that we have 50 kHz, we have such a high sampling frequency, every sample, every voltage trace can be 20,000 to 100,000 observations. And at most, we have thousands of samples given how many cells we are. We directly run into a classic data science problem of the features is way more than the observation available. None of your linear methods, it will be a yield post system to run dimensionality reduction directly. However, once we are able to survey a lot more cell instead of individual cell, the global classification results based on these features like resting potential and sag ratio actually is not satisfying. The accuracy is merely 45%, and from the previous result of the Greg of the Schwartz lab, they're saying that some of the sample is actually below threshold to be classifiable. So we want to find a better way to use this resolution. And that's actually another notion of the importance of using this type of intrinsic electrical properties. So intrinsic electrical property traces the cell's activity throughout development. And this resolution levels transcriptomics to provide us as possible characterization of diversity as possible. And the rest of the slides are going to be just raw results. And let me see. This first time I'm presenting it. So what we do is first, we don't know which time scale is available, which time scale is interesting. So we decide to apply self-supervised learning in a hierarchical way to apply to all of the possible scales. And all of the possible scales means all of the spatial scales and frequencies, temporal scale and frequency scales. And also we devise our architecture according to how many blocks of this self-supervised learning are necessary. And just by doing this, without us to provide label data, because as I showed you in the previous slides of self-supervised learning, it's contrasting between different variation of the sample. Instead, we tell them what labels are. Our results is actually double of what previously reported with other methods, especially given the classification, the distribution of the cell is not balanced. So we actually circumvented a lot of data science challenge from biasing the classification result, given the skew distribution of the data, as well as given the dimensionality, yield post dimensionality of the data. And second, we can also do the feature study, the feature analysis. What feature are necessary for what feature separates different cell types? That's unknown as well. So if we have 2 features, we already see 5 classes pop up. If we have 20 features, 10 of them pop up, but there are a lot more variations. And 200 allows us to start to map back to all of the ground truth cell types, and they actually achieve about 90% accuracy. And lastly, as we build out a scale, build back the hierarchy, as you've seen from the Cypher 10, we can do something very, very similar here to all of the subtypes as well as all of the raw types. So even given all of the cell type inaccuracy or the imbalanced distribution, we're able to recover the type hierarchy, the ground truth typology in the retina previously published in the Greg Schwartz groups. Okay. And because I'm a little bit short on time, I'll just say this is one, there are multiple, this is one of the ways of doing, I will skip this part. And this is one of the ways. And today we mostly cover the classic regime of using information bottleneck and open up to connect to AI. And second, the current work I'm doing, we're working on 2 different regimes of multi-sensory or multi-input comparison, cross-modality comparison of how the information bottleneck, when we relax that into Bayesian risk minimization, we can actually do not need geometric insight and still perform discovery of feature discovery. And third is the ongoing collaboration into improving biology or improving behavior. So that is a new work we just submitted with my colleague in Stony Brook. We are using this type of model-based analysis to predict what kind of experiment design would trigger or would be able to survey the response range or behavior range of D1, D2 strata-atom, which we know is critical for fear-based learning and for reward learning. And with that notion, I thank all my funding agency and I invite your questions. Thank you.
You showed in the last section that this method you developed gets back to ground truth cell types. Does the method infer new cell types that are unknown?
That's a very interesting question. I would say yes, but the new cell, but we define ground truth in the retina is because given different modality of the cell measurement, electrophysiology, morphology, and transcriptomics, we found converging cell types. And so this particular method, if I'm using intrinsic electricity, I would find new clusters. And whether that's ground truth could be rigorous. I'll need to compare it with other modality to verify that. But yes, we actually noticed that in when we applied it to Allen Institute's data set. Yeah.
Let other people go if other people have questions. So I have 2 different questions, but let's start with the first one just so I can understand the neural collapse thing better. You said it makes this equilateral triangle lattice type of thing.
Yeah.
So is there a metric for testing how well that works other than just the fact that you're test accuracy
Yeah.
gets really high, right? Like there should actually be like a number you can put on.
There are
how well
multiple
this
things you can,
metric
there
lasts,
are
right?
many things you can measure. Generally, neural collapse is 4 different elements together. First is your variance goes to 0. And second, that means intra-cluster versus inter-cluster, the proportion goes to small.
Oh, you
and-
can just get a number for it then. Okay.
Yeah, and then is the mean variance of the angle. And third is, that's the KNN. You test whether your linear declassification match your KNN. That's the ultimate test to say you are just finding the nearest cluster.
Okay, my second question is more of a philosophical challenge one, which is on the UNet thing and the sort of information bottleneck thing. It's a beautiful result, but the key assumption is that you know what the information that you're trying to extract is, right?
Yeah.
So for a moving bar, sure, the predictable trajectory of the next frame does sound like something that the retinal population would want to extract. Maybe that probably makes sense. You want to know where something is. The next frame of a movie, in terms of its actual pixel representation, I would argue is not what the retina
No,
actually wants to do, right? So like how much does that bias what you're trying to do, right?
that's yeah, that's true. How much of that bias what you try to do? So we actually, that's an encoder decoder. We're decoding on the movie frame, and one way you can do that is you're decoding multiple stream of future to not bias from one type of the temporal constraints, and the other way is you strip away the decoder. So you don't include the decoder information to bias that. You're just saying what's available in the neural code. That goes back to
Right, but still, I mean, the whole problem is set up for like where, right? You're trying to optimize, trying to decode where you are in a stream of a movie,
I think
which like fine, but like that's probably not what the cells are doing for the salamander, right? They're trying to like just not get it eaten or have it eat something, right? Like it's right, it's thing that it's trying to do.
So
Cost function
what we're
is different.
doing is a proxy of what's available, because what you can do is tell frame by frame what's the difference. And whether that's used, that's what you're saying here. Whether that's used, that's a different layer of question. If you know what's that used, then probably we can use a concrete format of the decoder function and replace the mutual information. Mutual information is a modern way to say what's available there
Yeah.
that can be used to discriminate different states. Yeah.
For the cell type classification, I'm wondering, what is the kind of input stimulus that is used? Presumably there are multiple. And second question is, can you kind of like back out what would be an optimal stimulus in terms of discriminating different cells? Like, is the chirp stimulus?
That's a great question. You guys looking at me. Yeah, so it's simply a set of current injections, right? It's steps of current injections. We certainly did it's just steps. It's like 12 different steps or 20 different steps. It depends on which cell. And they're like 1 second steps. This is a fairly common kind of paradigm, but that certainly doesn't answer the question of what would have been optimal, right? These experiments are hard. Like, should I have done more steps for shorter amounts of time? Is 1 second the amount of time to play each step? What are all the currents we should have injected? Because too small a current, it doesn't spike at all. Maybe that's useful, maybe it's not. Too big a current, it's saturated. Like, there's a ton of choices that can be made about that. And the short answer is I don't really know. Now that we've got this thing and we can actually measure its performance, I think one way to do that is just to like at least hold out some of the data, and Siwei started to play with that. So we can simply hold out the traces for certain steps and say, okay, we shouldn't use steps less than 50 picoamps. We can do that. We can't retroactively go and change the duration of our stimuli unless I run a bunch more cells. So yeah, other than kind of an iterative experimental process, I don't know if there's an easy way to get to that question, but it's a great question.
That's true.
What is the optimal thing to actually run in these experiments?
That goes back to the earlier question about we need some data set to benchmark this with ground truth. And so far, if ground truth means converging evidence of a cell type, then the only thing available is on retina. Like, for example, in Allen Institute, they published cell type data set. They have 17 electricity type, but then they have 38 morphology type, and then they have 46 ME combined types. And those are clusters. We actually don't know how they're relating with each other.
Any more questions? Let's send Siwei again.
Thank you.
movie that hard, like